if what you are publishing is material. And in the case that, in the study that Ingmar Riedel-Kruse and Paolo Blickstein have done, what they are publishing is biologically active substances. So a new twist on Publish on Demand, Professor Riedel-Kruse from Bioengineering teamed with Paolo Blickstein in the Graduate School of Education. Ingmar loves games. He's created a platform for biology games called Biotic Games. And in this experiment, they employ a kind of learning that is called hybrid learning. Ingmar.
Welcome. Exciting meeting. So my lab, one of the interests we have is basically how do we as humans can interact with microbiological processes. So we all see kind of these big plants and animals and whatever on a daily basis, but there's much more that we don't see, right? Microorganisms you have in your hand, in your gut, and then whatever. And so it raises the question, how can we interact with those? Should we? And so forth. And we are kind of interested in kind of making interfaces to do that, and then also apply them to meaningful applications. And so one of the things that you see here is kind of indicated is also this joystick. So as Martha said, we also made some games, but that's not what I'm going to talk about today. But what the two arrows signal is basically really want to have a two-way communication of some sort. And what I'm going to talk today primarily about is how can we make interfaces with the kind of devices we now increasingly have to run experiments, and primarily over the web. So that's what I'm going to talk today about. How can we do cloud experimentation? How can you basically sit in front of your computer and do experiments with microbiological systems? So why do we want to do that? There are multiple reasons for that, and just name a few. We talked a lot about like online education, big hype, and one of the things that is really missing is kind of real labs, right? I mean, you can't do experiments. And what we're interested in is how can we basically deliver biology experiments at large scale in these kind of educational settings. There are other things, like I personally come from physics originally. There are many people who do theoretical biology. They would like to do experiments, but they can't because it's hard to run a wet lab. But we know from cloud computation, you can actually have servers where you can do computation. Why aren't we doing the same with experiments? And then there's also a more practical aspect in the life sciences. We have increasingly more high throughput equipment, which is very expensive, somehow centrally localized. And why can't we open this up instead of one person running 100,000 experiments in one of these machines? Why not having 100,000 people running one experiment each? Right? Those are the kind of questions we ask. And what I want to tell you today is kind of some rather small-scale prototypes and what we can do with those, primarily in education. So the first example I want to talk about is about Physarum. It's an organism, a slime mold, which you see here. So this is a petri dish, for those who don't know. And kind of the scale, here's a couple of centimeters, and the whole movie is running about a day. And what you should see here is in yellow is this organism, which kind of grows in the dish, okay? And what you see in kind of grayish is basically where food has been dropped. And you can do this kind of experiments by hand, by pipetting by hand, and you see how basically this organism initially explores, but then over time kind of really follows this food. Now, the slight twist here is that this actually has not been done by pipetting by hand. It has actually been done online via a web interface. And how this basically works is we have some web interfaces, either via a phone or kind of standard web browser. We have a robot which we built out of Lego that basically has a bunch of these petri dishes and drops food on it. And what you get back as a user is your experiments. And I really sped this thing up. So this is the website where basically the students can log on, choose an experiment, then choose what kind of food and how fast they want to lay it, and they lay out a trailer food, might submit that, and then all this information gets sent to our robots, which are then basically execute as experiments. The reason we use Lego is twofold. One, it's easy to prototype. The other thing is, as was mentioned before, there's certain robotics competitions. So you could envision how kids in the future build these things themselves, right? And we have a scanner here which takes those images, like every 10 minutes. And then these images are basically put in the database, and the student can log on frequently, kind of look how the experiment is going, and put further food stimuli on there, right? So here again, the web interface, most students kind of use this one, but we made it such that you can also use it from a phone. You have some the main feature is you can really basically see your experiment, scroll back and forth, decide where you want to drop the food. We also have some zoom-in features, so you can really have close-up looks of how this growth is happening in small detail and so forth. We even had some chat features, so students could talk to each other. The robot, I said, is basically made out of Lego Mindstorm with a flatbed scanner, and the Raspberry Pi is kind of controlling the whole thing. So then we used this last quarter in a Stanford class that I teach in modeling multi-cellular systems. And so we had the students run throughout the class experiments. So each student did about 20 of those experiments. And here you see a bunch of those experiments that they have done. And what you kind of primarily can see is the different blue dots. You see what different things students tried. So a couple of those tried things like if you put two different blobs of food, where does the organism decide to go? Some students did very long trails, others tried all sorts of things, right? Different sizes. Other students didn't put any food, just watched. And so the students then did also final projects, so we asked them to model things. And this is kind of one example what one of the students did. Here's kind of the raw data looking at different sizes of these organisms, and try to capture what are kind of the different form factors that you get, kind of this fractal pattern. And then the student did quite some modeling of trying to think about different models that would be more or less realistic to what has been done, what is seen in experiment, and then also make some quantitative experiment. So the key feature here is really that the student can basically have a lab experience, but without having to actually physically doing any lab work, right? So another nice feature is that we can actually log all the user data, right? We know every click, we know when they zoomed in, whatever they did, and we can use this then to do learning analytics. And so here's one example of a snapshot of what such learning analytics looks like. So what you see here is the time axis, basically over this 10 weeks period of the course. What you see here is basically each experiment that were done successively. This is the data for one particular student. Each of these green bars is basically the duration of one experiment, right? And so you see basically these were the first experiments, 3 experiments in parallel, another 2, then there were done 4 experiments, and so forth, right? And then what you see kind of the ticks here is whenever the student logged on and did something, and the student did a lot, so we can really zoom in what they actually did there. And the blue bar usually goes until the last time they ever looked into this experiment. Right? What you can see here, for example, for this particular student is usually had this pattern whenever he did kind of a next run of experiment, he looked kind of at the last few experiments, but never really looked far back, also here, right? I mean, did these 3 experiments look at these, but never looked back at those. We also have kind of opened it up that the students can look at other students' experiments, so that's shown here. And you see that this student at some point looked at some other experiments, right? So this is kind of a snapshot of user behavior, which you could analyze in much more detail, which I can't go into detail, but just to show you the example for 4 different students, right? Kind of the same snapshot, and you should right away see some very obvious differences. For example, this was the student I just showed you, right? Which I just said usually did not look back at other experiments, while all these other students usually really looked often back at what they have done even weeks ago, right? Another feature is this student, for example, really looked a lot at other people's data. All the others did not as much. This student here really, I mean, as you see with all these purple graphs, really studied all his own experiments extremely carefully, right? So very different user behaviors. And the funny thing is, I mean, the students were graded, and we did the analysis after the course, and kind of what you see here also reflects a little bit the overall course grade. So kind of the student really put lots of effort in what you see here, put also lots of effort in other parts of the course. But so it's really powerful, not only for teaching, but also for learning research. I mean, you're still kind of analyzing that data. And I want to give you a second example of this kind of online experimentation system. So what I showed you so far was something when experiment basically lasts a day, and you get an image every 10 minutes. What about in interaction, which is kind of real time and which maybe lasts only a minute? And so the organisms we use here are euglenas, kind of single-celled organisms that are 50 micrometers long. You have them in your pond. So the interface we built here is that we have a chamber where you have all these organisms swimming, and then we have a joystick here which you can pull, and this joystick actually shines light, actuates light from a certain size. And if you shine light for a while, you should appreciate that then all these organisms move in one direction, right? And if you now change the direction of the joystick, then you can make the move in another direction and so forth. And so you have a real-time interaction with these organisms. And we didn't really make this into a game yet, but you can see here, for example, a box. We can say, like, try to get the box to the other side where the box kind of tracks the flow of these organisms. Right? So I think it's really apparent how they follow the stimulus. So here's the basic setup, the joystick, as I just showed you. The chamber is basically schematized here by 4 LEDs, which can be turned on and off kind of in an analog fashion. And that's what the thing actually looks in real. I'm not sure whether you get much out of it, but basically like a chamber of a size of about a centimeter. So very simple. The main thing here is that we've found an organism that really lives in this kind of chamber for weeks very happily, and we have a very robust and affordable system. Kind of the overall schematics is you can use a standard microscope, have this little chamber here, and then use an Arduino to kind of control it either locally or over the web. Okay. If you now do this just with different zoom levels on the microscope, right, you can look at very different features. This is what we saw. You can really look at high levels and see subcellular details. So I'm not sure whether this is visible on with the lighting conditions here, but actually the dirt particles in here, which kind of show you the whole flow field of the surrounding medium, and you can even detect Brownian motions. There are lots of details what students could study and discover while interacting with that kind of interface. Right? And there's also all sorts of other things that grows in there. It's kind of a little micro ecosystem. So there are other organisms you can discover. For example, multi-cellular. If you zoom in, you can see bacteria and kind of these tracer particles that are also mentioned. So very rich experience, and this is kind of the next thing we're going to use in another class. And so let me come to the conclusion here. So what I talked to you about today is we're interested in kind of human biology interaction. How can we enable people, all ages, to interact with these microscopic systems? The primary thing today was about doing this over the cloud, but we can also do it locally and with other kind of approaches. The two different things here this was really kind of a long-term experiment lasting a couple of hours, while this one is really kind of quick pace. And the applications for this, there are multiple ones. Teaching and learning analytics are those that I try to highlight today, but in the future we can also think about life science research. So, for example, some theoretician logging on, running their experiments, downloading data and analyzing it, maybe in the future even open it up for citizen science. Okay, that's what I wanted to say. Thank you.
Very much. Thank you. Thank you.